Okay, good afternoon, everyone, and welcome to the third annual NICoE Research Fair. Thank you very much for joining us this afternoon, and I'll get going by way of a couple of introductions and maybe kind of an overview of a bit of the history of the fair, the tone for the fair, and our framework for today. As would be no surprise to anyone, we're doing this fair in a different environment today than we would have just a couple of years ago or even 6 months ago. So I very much appreciate everyone's flexibility in navigating different virtual platforms and registering for this event. My role here at the NICoE, I'm Department Chief for Research. I've been here for about 3 years. And I think what I would say in terms of history is that when I arrived here, there was a general consensus that there could be some kind of a forum for this kind of an event. And no one really had a sense for what that might look like. And so as the years have gone on, the concept for this has evolved. And we'll talk a bit about the frame for today in just a bit. The general tone and the reason for this fair to come about really had to do with this awareness about MHSRS and this idea that not everybody could go. And so this whole idea of bringing back information learned in a way that would be meaningful to everyone, researchers certainly, but clinicians, admin staff, house staff, really anyone who wanted to learn a bit more about what was going on in TBI science. And so we started this fair concept with this idea that we could create a tone of accessibility, not informal, but really kind of intentional, so that we could welcome really anybody into the conversation to ask questions, participate, and feel like you could leave an educational offering with knowledge that you didn't have before, and maybe a curiosity and interest that weren't present before. So the frame for today, we'll have a plenary speaker for the first hour, and then we will have our first try at this, which will be a series of brief presentations over the course of an hour. And we'll see how that goes. Everyone's going to be on their toes and flexible, and we'll see how this technology holds with that. In the third hour, we'll have a presentation from Ms. Sarah Kruger having to do with the 10-year history of work in the caring. So we look forward to that. So without further ado, I will introduce our plenary speaker. We had talked a bit about how to do the bio for Dr. Anjan Chatterjee, and he seemed comfortable with me saying something like he's just an overall good guy. But then there's a bit more to the story there, so indulge me as I expound a bit. Dr. Chatterjee is the Frank A. and Gladys Elliott Professor and Chair of Neurology at Pennsylvania Hospital, founding director of the Penn Center for Neuroaesthetics. He's a member of the Center for Cognitive Neuroscience and a member of the Center for Neuroscience and Society at the University of Pennsylvania. BA was received from Haverford College, an MD from the University of Pennsylvania, completed his neurology residency at the University of Chicago. I'm also present to research fellowships that were completed at Case Western and also University of Florida. He is or has been on the editorial boards of a number of journals, American Journal of Bioethics, Neuroscience, Cognitive and Behavioral Neurology, Neuropsychology, and many, many others. He was awarded the Norman Geschwind Prize in Behavioral and Cognitive Neurology by the American Academy of Neurology and the Rudolf Anheim Prize for Contributions to Psychology and the Arts by the American Psychological Association. He's the founding member of the Board of Governors of the Neuroethics Society and past president of the International Association of Empirical Aesthetics and past president of the Behavioral and Cognitive Neurology Society. With that introduction, Please welcome Dr. Anjan Chatterjee for our plenary talk.
Trevin, thank you very much for that terrific introduction. The first line would have been sufficient. So I have received permission to take the mask off while I talk up here at the podium. I think in part it makes it easier for people listening. We all read lips much more than we probably anticipate, although many of you have probably realized this during the time of the pandemic. Having said that, if there's anybody in the room who is uncomfortable with me taking my mask off, I'm happy to keep it on. So seeing no objections. Okay. The other thing to point out is that this is the first time in over 6 months that I have been out and giving a talk to a live audience. So I'm a little out of practice. So be patient with me if I don't get all my transitions right. You might wonder why, in a talk about aesthetics, do I start with an image of such a dour-looking guy? It turns out this is a man named Gustav Fechner, who lived in the latter part of the 19th century, and he's really critical to everything that we are doing right now. And there are 3 points that Fechner made in his career. He's regarded as one of the founders of experimental psychology. But there are 3 points he makes that are relevant to what I'm going to talk about. The first, and it is claimed that in October 1850, so this is 170 years ago, he woke up with a kind of epiphany, and the epiphany was that properties of the world are systematically related to properties of the mind. That somehow if we think about the world and we think of the mind as separate entities, it doesn't quite work. And that the way he didn't use evolutionary terms, but the way our brains evolved, that there are systematic ways in which the world is related to the mind, and this can be modeled mathematically in a way that came to be known as Fechner's law. His second claim was, if this is actually true, that properties of the world systematically map onto properties of the mind, this had to be mediated through our nervous system, and recognized that in the late 1800s that the technology did not exist to be able to investigate this. And so he claimed that in the same way that there was an outer psychophysics, which is his thing about properties of the world, that there had to be an inner psychophysics, which is that properties of our brain were systematically related to properties of our mind. And that essentially is what much of cognitive neuroscience is trying to understand what the physiologic properties of the brain and our nervous system, how those are related to our mind. And then the third piece relevant to this is that he was the first one to really push this idea that aesthetics could be an empirical science. And he called this aesthetics from below, and this was portrayed in contrast to aesthetics from above, which is what had been done for most of the history before this, which is people in the humanities, philosophers, art historians had sort of talked about the nature of aesthetic experience or aesthetics using first principles and logical arguments. And Fechner was the first person who said, well, we can actually do experiments to see if these ideas actually bear out. And so these 3 things, I think, make up the core of neuroaesthetics, which is that properties of the world are systematically related to properties of the mind. They're mediated through our nervous system, and one can have a programmatic approach to aesthetics using experimental methods. So it should be clear that neuroaesthetics is a very young field. And just to give you a sense of that, this is from a PubMed search I did. looking at publications from 1965 to 2019 with the following search terms: neuroaesthetics, or neuroscience or neuropsychology in art, neuroscience or neuropsychology in beauty, and neuroscience and aesthetics. And you can kind of see the trajectory of publications. Essentially, before 20 years ago, there was virtually nothing. And then it has started to pick up. And really in the last decade, maybe 10 years, 12 years, it's really ramping up. So this is a new and burgeoning field. And part of the reason for bringing this up is that it is often the case that people, when I give public talks or talks to other academics, people will ask perfectly reasonable, good, insightful questions, and more often than not, my answer is, we don't know, because this is a field where there are many open, low-hanging fruit kind of questions that have just not been addressed. And for students, I like to tell them that this is an area where anything you do is going to be a contribution, because it is so young at this stage. But to set the frame a little bit, this is, when we think about the domain of inquiry, it's worth separating out the cognitive neuroscience of aesthetics and the cognitive neuroscience of art. And this will be fairly self-evident. You can certainly have an aesthetic experience of natural objects like landscapes and people that are not necessarily art, and vice versa. And within this domain, the cognitive neuroscience of beauty, which can apply to natural objects as well as artifacts, has probably received the most attention. But as the field is evolving, it's becoming clear that aesthetics is more than just beauty. And how these other kinds of aesthetic experiences are accounted for has become more of a consideration in the field. This is a general kind of model that we had proposed a few years ago to kind of organize the thinking. And the idea is that aesthetic experiences are really an emergent property of a number of different large-scale systems. So this includes our sensory motor systems. And to give you a trivial example, which is that during a sunset, we do not have an aesthetic experience of the infrared spectrum of the light. You know it's there, you know it's doing something, but the design of our biology is such that we do not apprehend that part of the spectrum of light, and so there is a way in which the design of our sensory systems and our motor systems constrains the conditions in which we can have an aesthetic experience. The second is this notion of the emotions and valuation piece of an experience. So often people think of aesthetics as really a kind of valuation, right, of an object, fundamentally. And then the final thing is that all of this gets modulated by knowledge, by meaning, by people's personal experiences, which affects this axis from our sensory motor systems and our emotional systems. And as a simple example of that, again, if you ask most Americans what kind of art they like more than others, the most common response you get is Impressionist art. 150 years ago, when Impressionist art first came on the scene, nobody understood it. It wasn't that the Paris salons rejected them. It was really regarded as something bizarre. Our brains really haven't changed in 150 years, but the culture in which we exist and the kind of exposure we've had to this kind of art has changed dramatically. So people often talk about, how variable is our response to beauty? Isn't beauty in the eyes of the beholder? And my response is typically that beauty is really in the brain of the beholder, and our brains are more similar than they are different. But it's this axis around knowledge, meaning, personal experience, your cultural upbringing is where most of this variability comes in. So with all of that as kind of background information, let me delve into some of the work we've been doing. And at my center, we are especially focused on the visual system. and the way in which the visual system contributes to aesthetic experiences. And you can imagine every sensory modality has access to aesthetic experiences, the most obvious being audition and music, but every other sense as well. You can think about smells and perfumes, taste and sort of the quality of an eating experience you might have, touch, with tactile experiences of fabrics. So you can have aesthetic experiences through every sensory modality, but we're focused on vision. And if you think about the visual system in our occipital cortex, the visual world is basically carved up into people, places, and things. And so we are interested in the experience of all of those. What does it mean to have an aesthetic experience of another person, a place, which can be landscapes or architectural built environments, and things can be the design of objects or the prototypic object that we think of aesthetic experiences, which is art. So I'm just going to touch on each of those a little bit. So when we think about faces, or when we think about beauty, typically people think about faces. You think about someone being particularly beautiful. We're often imagining what they look like. We were interested in what the biologic basis is, or what is the biologic account for these kinds of experiences of beauty. And in the early aughts, there were a few papers that had started to look at this, and the general, which I'll talk about in a minute, the general conclusion is that the experience of beauty seems to be the simultaneous neural activity within our visual system, in occipital cortex, and within our reward systems, deeper in the brain, in the anterior parts of the brain. When you have both of those areas being active simultaneously, that is an account of what the experience of beauty is. We looked at this. This is a paper that was published in 2009. It was an fMRI study, and this is where people came in two different sessions. In one session, they saw a series of these faces. These were all computer generated. They're not faces of real people. And in one session, they made an identity judgment, which is that they looked at an image and said, yes, I've seen this person before, in the series of images that they were being shown. And in another one, they said, they made a judgment of, is this face attractive or not, right? So they're looking at the exact same faces, making two kinds of judgments on different sessions. And this is to show what we found, and I'll direct your attention to the left panel and the bottom three images, which is, if you look at the left part where there's blue, red, and yellow parts of the occipital cortex, and just to orient everybody, we're looking at the back of the brain underneath it, so if you're looking up the brain. And what is shown in yellow is part of the fusiform gyrus, which is known to be sensitive to faces more than other kinds of things. The red is part of the parahippocampus, which is more sensitive to places and buildings than other kinds of things. And in blue is shown an area called the lateral occipital complex, which is generally regarded as more sensitive to objects over scrambled images of objects, right? So this we know, and we functionally identified those separate from our experimental task. In the middle panel, what you see is the response when people are making judgments of beauty, and the way in which the neural response varies parametrically with how attractive the face is. And so what we find is that there are parts of parietal cortex, some of medial frontal cortex, and then parts of the insula, and then this area within occipital cortex, which is what I will focus on. What this is saying is that part of our visual system is responding to beauty, but it turns out not in a generalized way, because the part of the brain that is responsive to places, the parahippocampus, is not responsive to how beautiful these faces are. Right? So it's not a generalized visual response. It's specific to faces and objects. Now, it's not obvious that this is how it would play out. For example, if I told you the design of the nervous system is that our visual system classifies the world, it says this is a person, this is an object, this is a place, and the front of the brain, where our reward systems are, does all of the valuation, this would seem like a perfectly logical way for the nervous system to be organized. But that's not what we find, which is we find way back in the visual system that this evaluative function seems to be happening. So very early, and I'll come back to that in the processing. What's even more striking is the bottom panel to the right, and that's the identity condition. So this is where people are not being asked about beauty. There's nothing about beauty that is explicitly on the table. They're just looking at a face and saying, is this a familiar face from what I've seen before? Under those conditions, we get the exact same response in occipital cortex to the beauty of the faces, to the same degree as when people are making an explicit decision. And this suggests that this part of the brain is responding automatically to beauty even when people are explicitly engaged in another condition, right? And so it seems as though these evaluative responses are happening, at least in the case of faces, and maybe it generalizes to other things, but are happening automatically almost all the time, regardless of what explicitly people are thinking about. So the next question is, so here's another paper that was not from my lab, but was done another group around the same time that really focused on the reward parts of the brain. We at the time did not have good signal in that region of the brain, and what they found is within the nucleus accumbens and medial orbitofrontal cortex, these are classic reward areas, that they also found these automatic responses to beautiful faces when people were making a perceptual judgment task rather than an evaluative task. So we asked the question, is this kind of automaticity, automatic reaction to beauty, does that get expressed in behavior in any direct way? This is a simple study we did with collaborators in Singapore, and it's a straight-up behavioral experiment on a computer, and the task is the following. People see this kind of screen. There's a mouse that's pointing to the number at the bottom, and their task is to move the mouse to whichever of the numbers at the top right or the top left is closer to the bottom number. All they have to do, so 41, so that means that they would, move to whichever number is closer, and the faces that are next to the number are completely irrelevant to the task. People do this easily, they're at ceiling, people don't make errors. What we were interested in is what is the effect of the face that is at the distractor location. Does that have an effect on motor behavior? That's the question. And this is what we find, which is that when people move to the closer number, but the attractive face is in the opposite side, unbeknownst to them, their hand moves slightly in the direction of the attractive face before it lands on the correct number. And they're not aware that this is happening. And this suggests that when we talk about attraction in faces, this is not simply a metaphor. This is our motor systems are actually being engaged to actually move in the direction of the attractive face, even though the task has nothing to do with the faces, right? The performance of the task. Again, arguing in one way that our motor systems are being deployed automatically to faces that are attractive. The story ends up being slightly more nuanced because we also did eye tracking in these participants. And I'll just have you look at the panels to the left, which is the one at the top is the one around limb movements. But the bottom one, as you can see, kind of shows you a U-shaped curve. And one detail about the experiment, which I didn't mention before, is we had taken a whole bunch of faces and had them rated and took the top 10% as attractive, the bottom 10% as less attractive, and in the middle were what were regarded as neutral faces. The less attractive faces did not have an effect on the limb movements, but it did have an effect on the eye movements. And what we find is that the eyes move towards, as far as initial fixation, and stay longer for duration at both extremes, at the attractive faces and the less attractive faces as compared to the neutral ones. So what this is telling us, or at least our interpretation of this, is that the same information is coming through our visual system to our occipital cortex, and then it gets deployed to our motor systems, our output systems that are doing different things, which is that our limbs are moving in a way that suggests attraction to the beautiful faces, but our eyes are doing something different, which is that they seem to be vigilant to outliers. It doesn't matter if it's a very beautiful face or an unattractive face. What our eyes are doing is really paying attention to anything that's going to be an outlier of the norms. And so here's an example of an automatic response to visual beauty in faces in which our motor systems are being engaged in different ways. Okay. Now you can think of at a higher order of behavior and automatic responses to behavior. It turns out that social psychologists have identified this to quite an extent, which is called, sometimes referred to as the beauty is good stereotype. And attractive people receive all kinds of unearned advantages in life. Turns out, people who are attractive are hired more easily, they're given more pay. If they commit infractions, they're given less punishments. Children that are attractive, unless they're standardized tests, are given higher grades. And so there's this whole world out there of privilege that has nothing to do with gender, race, any of that has to do with how attractive you are. And people who are attractive have all kinds of privileges is something that has not been emphasized very much. So we got interested in this, this whole idea of valuation, of what is the nature of beauty valuation and moral valuation, and what are the consequences of that. And here's one example of an imaging experiment. Not done by us. This was done in South Korea. This is where people are looking at faces and making judgments of the beauty of faces again. And they're also given these little cartoons. And the cartoons represent an event that is either a neutral event or it's what most people would regard as a morally good act. In this case, it's the morally good act is a young girl helping a blind person down the stairs. Most of us would think this is a good thing to be doing. So what they find is within orbitofrontal cortex, which is, again, an important part of our reward and valuation system, that there are areas where there is convergent neural activity for both moral acts, a judgment of moral goodness, and a judgment of physical beauty. And it suggests that this might be a biologic tether, or this kind of thing might be a biologic tether for this, this stereotype of of the beauty is good stereotype. And this is only one. I think by now there are about 4 or 5 papers that have shown something along this lines. We decided to approach this in a slightly different way, which is, again, because I'm in a school of medicine and we have a very active surgical program, and now some of what I'm going to talk about will be directly relevant to, I think, some of probably the vets that you guys treat, especially vets that have had facial injuries of various kinds. But in this case, we looked at people with various kinds of facial anomalies. So we combed through all these surgical texts to find enough images where we had a pre- and post-surgical image, which were taken in relatively similar ways, similar lighting. And this was a grab bag of anomalies, so skin cancer, developmental abnormalities, Sturge-Weber, Port-Wine-Nevide, these kinds of things. We wanted to know is whether there was an unfortunate complement to the beauty is good stereotype, is there a facial anomaly is bad stereotype? And this is what we end up finding, which is that people respond to faces with these kinds of minor anomalies where they are making the inference that the person is less competent, less honest, less trustworthy, less intelligent, and so on. That without even realizing it, this is what people, when you see someone with this kind of anomaly, that is a typical response. And so when you're thinking about, I know sometimes in this world people refer to hidden injuries of soldiers. You can add this to one of the hidden injuries that soldiers that have minor facial wounds when they've been out and deployed, that they are now, even people who know nothing about them assume that they are less intelligent, less competent, less trustworthy, less honest. This as a stereotype gets really reinforced in popular media. And just to give you examples of, whether you think of villains from the Marvel universe or for Bond movies, that it is, and once you start paying attention to this, you'll see this all the time, that as soon as someone is supposed to be bad on the screen, they're often given some kind of facial abnormality, and you don't have to build up a big backstory of the development of their villainous character. The audience knows right away. And we do this very early. So The Lion King, such a popular movie. The villain in The Lion King is the only character in the entire movie that does not have a name that is other than his deformity, Scar, right? So our 3-, 4-, and 5-year-olds are being taught this message very early on. So we took the same idea and looked at, this is the implicit association test, and this, many of you may have heard this or perhaps done this. This has been used very commonly in both race and gender studies. The general idea is that you look at a face, you have to decide, is the face a typical face or anomalous face by responding in one direction or the other. And very easy task. And interspersed are words, and you just have to respond, is this a positive word like happy or a negative word like pain in the same direction. Again, very easy task. The key is that if there is an implicit negative association with a specific kind of object, in this case anomalous faces, that the responses will be quicker if both of those are categorized in the same direction. So if I have to respond to the right to say this is an anomalous face, to the right to say that this is a negative word like pain, if I'm moving in the same direction, it's as though our motor systems are primed, if that association exists, to respond faster, right? If there's no association, there shouldn't be any bias in one direction or the other. So that's the basic logic of this kind of experiment. We also developed an explicit bias questionnaire, just asking people how much, a series of questions, basically saying, do you really think you're biased against people who have facial anomalies? And what we find is that in the explicit questionnaire on the left, that the people's responses as a group are no different than 0. So as a group, neither men nor women claim to have an explicit bias against people with facial anomalies. But on the right, if you look at the effect size on this implicit association test, the effects are quite robust. It turns out, and we've replicated this, these effects are more common among male participants than among women participants. So it looks like men generally are more biased to these kinds of facial anomalies than women. And if you try to correlate their own responses to their self-awareness of explicit bias and the effect sizes of their implicit bias, there's zero correlation. And so the big picture around this is that I would submit that everybody in this room has some kind of implicit bias. It might not be for people with facial anomalies, it might not be for any group you want to talk about, but I would submit that everybody in this room has a set of implicit biases that we bring with us, and we are the poorest judges of our own biases, is the implications for these kinds of results. So now we take this to the scanner to ask the question of what's the neural basis for these kinds of effects. And this is where people, again, are looking at these faces. All they're doing is making a judgment of, am I looking at a man or a woman? So there's no evaluation here. And there are two main things I'd like to point out, which is that if you look in occipital cortex, this is in the bottom left of that panel, there's increased neural activity to anomalous faces as compared to typical faces. And it turns out that this is in the same area as that first study I showed you in which we talked about automatic responses to beauty. And so this has caused us to revise our thinking, because back in 2009 we thought maybe we have automatic beauty detectors, but if that's the case, we shouldn't get this pattern here. So right now we're thinking, and I'll show you again some more data, that in the same way that I was talking about eye movements, that our occipital cortex we think is responding to the salience of images. So you have things on the extremes, and that's what is happening. We don't know if this is an initial response or this is a feedback response. And so when Dr. Braga was showing me his MEG information this morning, some of those data where you can really see the flow of information, this is the kind of experiment that to me would be fascinating to see if this occipital activation precedes or succeeds what's happening more anteriorly, which fMRI just doesn't have the temporal resolution to address. The other piece is in the middle panel where you see the green, there's deactivation, and this is an area that has been associated with empathy, and some people have suggested that maybe this is a biomarker for dehumanization, that is part of what's going on automatically. So the next experiment in this line is probably our most ambitious experiment to date, which is currently under review, where we try to disentangle a bunch of these phenomena with the basic question of what does it mean? What is this construct of dehumanization? What does this actually mean? And can we try to deconstruct that? So the experiment in this case, again, it's an imaging experiment, is where people are seeing a series of faces. They're habituated to a particular small set of men and women faces, and interspersed is a novel face, right? So, and the novel face can be one of three kinds. It can be a beautiful face, it can be an anomalous face, or it can be an average-looking face. It's just a new face, right? And what this allows us to do is ask the question of, for salience, the general notion is that a salience response is to anything that's novel. It doesn't matter if it's beautiful, ugly, if it's whatever. As long as it's novel, that's going to be salient, right? Our nervous system is designed to attend to things that are novel, right? Separate from that, maybe there's a beauty response, which would suggest that the response to beauty would be greater than average, then greater than anomalous. And then the third possibility would be a kind of U-shaped, which is respond to the extremes, which might be an emotional salience response. So just to clarify the logic of this, and we looked at a number of other things, which is we had these implicit association tests on people. And another thing we looked at is wanting to know if people have these kinds of negative biases, does this cash out into behavior? Because if it doesn't affect your behavior, who cares, right? Whatever you think in your head, who cares? As long as it doesn't express itself into the world, you know, have at it with your negative implicit biases, right? Nobody cares. So we wanted to start to look at that. So this is there's a lot of stuff in here, and since this is not has not been accepted for publication, it's under review right now. I'm only going to point out a couple of things, which is at the top left, if you look again at the bottom of the brain, those same areas that I've shown before really seem to respond to salience, which is as soon as something is out of the norm, is novel, that's where the response is, right? So we can be fairly certain about that within occipital cortex. We also separately identified parts of the brain that respond to disgust, and the disgust is either to animals that are that people find disgusting. So, you know, snakes and rats and that sort of stuff. So we're not talking about your, you know, your fuzzy kitten, you know, those sorts of animals. And to bodily injuries, right? So different kinds of disgust. And one of the areas that in kind of affective neuroscience is gained some importance now is that there are different forms of disgust, and how do those play out in these other contexts. So the only thing I'm going to point out is in the larger slide is the left amygdala seems to be a critical place for a number of things that are happening, which is that in the left amygdala, the neural responses vary by the degree of effect size on the implicit association tests. So how much of an effect you have with these implicit biases gets cashed out in neural activity in the amygdala. This is also an area in which we identified that it's specifically animal disgust, not bodily injury disgust. Again, consistent with this idea of a kind of dehumanization happening. And then we used some of these techniques from behavioral economics, or something called the dictator game, which is a way in which you get a sense of how much resources are people willing to share. And what we find is people vary on this. And so some people are much less generous if they're looking at someone with an anomalous face. And we find that that variability is also cashed out in neural activity in the left amygdala. So the left amygdala seems to be doing a lot of this work around biases, negative implicit bias, again, negative implicit biases, a kind of disgust, and it appears to be also having an effect on how people behave in these very controlled paradigms. And so this is just the beginning of this. You know, hopefully I have an NIH grant that's going to be reviewed in 3 weeks, really to try to explore much of this. But that's where we are right now in this line of research. But again, what I want to point out is for those of you who are treating veterans that have had facial injuries, because based on them, you know, whatever kind of IED exposure they've had and that sort of thing, this is something to keep in mind in addition to the physical injury that they've experienced, that the way they are being treated by other people is an added burden that they you know, that they bear. Okay, so I'm going to move on now to architecture. How am I doing on time, by the way?
15 minutes.
15 minutes. Okay, so this is what happens when you're out of practice giving talks. So I'm going to move through this quickly and come back to art. So as I was mentioning as I was coming in here, how notable it is that this particular physical structure we're in is so organized in a curvilinear fashion when some of the other buildings on the base are very rectilinear. And it turns out there's a lot of evidence for the idea that even going back to primates, there's a preference for curved shapes over rectilinear shapes. So whatever you're doing in designing this, I think, comports with some of these empirical aesthetics findings. I am going to skip this. So a few years ago, we started this line of research of asking the question, a very basic question of, is our response to architectural interiors similar to the other kinds of reward responses we have? It's a very basic question. And we looked at images, and these images were chosen, curated by a couple of architects in Denmark, where we varied along 3 dimensions, which is, are ceilings high or low, are interiors rounded or squared off, and is the space open or closed? And in this case, open means you can see past the space into whatever is behind it. And the finding at the time was, the relevant finding for that paper was that parts of medial orbitofrontal, ventromedial prefrontal cortex was responding to the beauty of these spaces. And we just wanted to make a very simple point that our responses to beauty in architecture affects our reward systems in a way that is similar to what has been seen in other domains, right? So that's almost a prologue to what I'm going to talk about. What we did later was, more recently, is we combed the literature for the kind of psychological dimensions that people talk about in response to architecture and came up with this list. And here we took the same images we had before. So there are 200 of these images. We did an online study of 800 people to look at how they rate each of these images. And when you do that, you can get this kind of matrix. It's a correlation matrix where certain of these 16 dimensions correlate very highly with others based on these spaces. And so this allows us to kind of carve out this dimensionality. So we use 2 techniques, 2 data reduction techniques of, like, the general question being, how do you take this complex domain and reduce it to something a little bit more manageable? And one is called semantic network analysis. And I won't go into the details of that, partly because I don't understand the math behind it. But basically, this is a way to say, if certain of these dimensions are, images are being rated very similarly across these dimensions, those end up being close in space. And this is a part of a movement in cognitive sciences now called network science, where you can create these kind of spatialized networks of the data. And what you see is you get these 3 kinds of responses, which we are naming coherence, how coherent is a space; fascination, which is the sense of you feel like you want to explore the space, there's something intriguing about it; and hominess, which is you come into a space and you feel comfortable. You feel like, yeah, I can be here, I belong here, right? And these things are orthogonal to each other. So you can be high on one and low on the other across the board. And most people have an intuition about hominess, so we've all been probably in very well-appointed hotel rooms that are, quite coherent. They can be fascinating, but you just don't feel at home in them. We also did a more traditional principal components analysis on the same data and basically confirm the same 3 components. And then we replicate this again, find the same 3 components. So we feel fairly confident that these components are important. So now what we do is we go back to the original study that I showed you that was published in 2013, because we used exactly the same images, a different population. That was done in Tenerife, Spain. These data were collected from Philadelphia, but online through the US. So again, different population. But now that we had identified these principal components, we could go back to their imaging data and remodel it in a way that so this is about as good of a double-blind experimental study as you can have, because when we were doing the study, we hadn't discovered these components. We didn't know to even ask the question, and certainly the participants didn't know that we were interested in this. And what we find is that within occipital cortex, there is neural responsiveness to each of these components. Again, arguing that this idea of fascination of hominess and coherence probably has some biological and psychological reality of how we respond to spaces. And I would just suggest as a kind of informal exercise, in the next week when you're in new places, just ask yourself, does this feel coherent? Does this feel fascinating? Does it feel homey? And you'll start to get a feel for how this stuff plays out once you make it explicit. And so right now we're taking this and trying to understand how this applies in real spaces. Can we generalize this? I've been involved with an architecture firm trying to design, thinking about designing of memory care units where we try to use some of these ideas in thinking through design. Okay, now I am going to, should I do art or should I stop? I should? Okay, so I'll zip through it. Question is, how do we engage with art? What is the nature of engagement with art? And there's been one idea, there are various buzzwords that go around like embodiment and enactment, and this idea that, our motor systems are somehow involved in artwork. So we got interested in whether our experience of art in which there's a lot of movement, so Jackson Pollock being the prime example, somehow engages our motor systems. And as a contrast, we wanted to look at Mondrian paintings, which are relatively static. So the advantage is these are all abstract art, so it's not about the representation per se. It's really how much movement is there within the abstraction. And this is a study we were going to do in Parkinson's patients. Again, the idea being that if you have a motor disorder, how does this affect your engagement with art in which there is movement, is the basic research question. So we took a bunch of Mondrians and Pollocks and basically confirmed this is an norming study, we confirm our intuitions that Pollock's people experience a lot of motion in them, and with Mondrian's not very much. If you look at the correlations, so we took 43 people with Parkinson's disease and 40 controls, and you can see these kinds of evaluations like beauty, interest, and liking all are highly correlated in these populations. For theoretical reasons, interest and beauty can dissociate, but in this case, they didn't. But here's this and the next slide are really the money slides, which is that if you look at the top row where motion, the amount of movement, is the independent variable, what you see is that people with Parkinson's, in terms of their liking, they seem to hit a peak. They don't hit the same peak as the control population, right? So they are being less responsive in their liking to the amount of motion in the artwork. But it's not the case that they just don't appreciate art in the same way, right? Which you could imagine that this is a dopamine deficient state. Maybe their reward systems are affected more generally. Because if you look at the relationship to the complexity of the art and liking, they're indistinguishable from normals. So it's one variable, complexity, that they're fine in terms of their aesthetic appreciation, but specifically when it comes to motion, that there's something off with them. And this is basically another way of showing the data, which is that the normal controls seem to have this kind of slight U-shaped curve when it comes to motion, in a way that the individuals with Parkinson's don't. And we actually think, if you look at the data in detail, is that the whole the curve of the people with Parkinson's is moved to the left, so it sits on top of just the ascent portion of what normal elderly controls are doing. And so the claim is that the aesthetic experience in people with Parkinson's is altered specifically in the parameter of movement in a way that it's not altered for other parameters. And it's one way for us to try to dissect the different components of an image that contributes to an aesthetic experience. And just to highlight, and this one is also under review right now, but just to make the point is, trying to understand what is the nature of engagement with art is really the question that leads into the bigger question, which is what you guys are all dealing with, which is what is engagement with the arts good for? Right? That's really the question, the ultimate question we want to get to, which you as practitioners are already doing it. And right now we have some funding from the Templeton Foundation to try to ask the question of what kind of transformation can occur in people who are engaged with the arts. And that in the next year is going to be a big focus of what we do. So sorry I ran on a bit, but most of this is new data that I'm excited about. So thank you for your attention. Should I take questions, or do we have time? Okay.
Thank you so much, Dr. Chatterjee, for your plenary presentation and for sharing your time and expertise. I know I, for one, will be taking a number of different nuggets from your conversation, not the least of which is really thinking about implicit bias when it comes to working with patients who have facial abnormalities, for sure, and more. Much appreciated. Everyone on the line, I know we have a number of people on the line and a small footprint of people here as well. We're going to take about 5 minutes just to take a quick break, do what you need to do. We have a number of presentations that I know are mobilizing and getting ready to participate in the next phase of this fair. But again, thank you, Dr. Chatterjee.
Hello, my name is Sarah Limba, and I am a research investigator at NICoE. I was previously a research neuropsychologist with the DIVBIC 15-year longitudinal study, and today I'll be presenting data from that study. So this abstract focuses on identifying acute predictors of future neurobehavioral symptomatology and postconcussive syndrome. Can you move the slide forward? Thank you. So as I said, this data comes from the 15-year longitudinal TBI study. In order to be included in the current analysis, participants must have had baseline data collected between 0 and 8 months of their injury, and also a follow-up evaluation conducted 2 or more years post-injury. Additionally, we excluded anyone who failed the Validity 10, which is an embedded symptom validity indicator within the neurobehavioral symptom inventory. We had 4 groups of participants: non-injured controls, trauma controls, uncomplicated mild TBI, and complicated mild, moderate, severe, penetrating TBI, all lumped together in a final group. And this slide just shows their demographics, military characteristics, and time since injury. And non-injured controls were older and had more years of education than the other 3 groups. They had more deployments than the uncomplicated mild TBI group, but otherwise there were no differences in terms of time since injury at either time point, sex, or race. Next slide, please. So this slide shows the potential predictors that we entered into the stepwise regression models. Demographics, military characteristics, each of those 4 injury groups, and baseline symptoms were all entered as potential predictors. And there were 2 main outcome variables of interest, which were the NSI, or Neurobehavioral Symptom Inventory total score, and whether or not the participant met ICD-10 criteria for post-concussion syndrome. So all of our models included several potential predictors. Next slide, please. And so these are the results of the stepwise linear regression. So again, we took all of those potential predictors and used a stepwise model so that the computer chose which were the final predictors entered into the model. And this is predicting NSI total score. And the first predictor that got entered into the model was NSI somatosensory symptoms. And that explained 48% of the variance in remote post-concussion symptoms. This was followed by cognitive concerns, PTSD symptoms, participants with complicated, mild, or more severe TBI, and sleep disturbance. These results were a little surprising to us. We had expected that PTSD symptoms would explain most of the variance in future post-concussion symptoms. So we took a look at the shared variance among these predictors, and we found that indeed the majority of the variance was shared by these self-reported psychological behavioral health symptoms. So 52% of the variance in future post-concussion symptoms was explained by the shared variance between cognitive concerns, somatosensory symptoms, PTSD, and sleep disturbance. And only a small amount of the variance was attributed to each of these factors individually. Next slide, please. So this final model was a stepwise logistic regression looking at the same predictors of post-concussive syndrome, future post-concussive syndrome. So here in the final model, cognitive complaints again, sleep again, somatosensory symptoms again, and injury group were all significant predictors of future post-concussive syndrome. And we did rerun this model, removing neurobehavioral symptom inventory predictors because we used the NSI to define our post-concussion syndrome groups. And when we reran these the model excluding the NSI items and clusters, PTSD did enter the model along with cognitive concerns and resilience. And both of these models, whether or not they included neurobehavioral symptom inventory items and clusters, classified 81% of participants correctly. So throughout all of the models that we ran, we saw that self-reported cognitive concerns, sleep, post-traumatic stress, and somatosensory symptoms were highly interrelated and were all strong predictors of future post-concussion syndrome and symptoms. And we did see that severity of injury impacted the likelihood of future symptomatology, but it was not nearly as strong as a predictor as the self-reported neurobehavioral symptoms. So overall, these findings suggest that patients reporting psychological distress and cognitive concerns acutely should be targeted for treatment to mitigate prolonged neurobehavioral symptoms. Thank you.
Hi, my name is Carrie Rosen, and I work in the Karen Lab with my colleagues Kathleen, Kyle, and Ms. Sarah Kruger, who will be presenting later. In this project, we did a retrospective analysis examining the relationship between conventional vestibular outcomes and immersive bell and task performance in service members with mild traumatic brain injury. Next slide, please. So just for a little bit of background, mild postconcussive symptoms can include vestibular impairments, such as chronic dizziness and postural instability, which can be compounded by the presence of psychological comorbidities, such as post-traumatic stress disorder. And during conventional vestibular evaluations, there's generally some subjective assessments, such as the Dizziness Handicap Inventory, or the DHI, and the Activity-Specific Balance Confidence Scale, or the ABC, while also some objective measures, such as the Sensory Organizational Test and the Functional Gait Assessment. And at the NICoE, we have the ability to incorporate the Computer-Assisted Rehabilitation Environment, or the CAREN, as you can see on the top right, to assist in the rehabilitation, but also look at their performance. And so the objective of the study was to explore the relationship between these conventional measures and their performance in the immersive balance task, as well as explore the impact of PTSD. Next slide, please. So on the bottom left of your screen here, we've got two separate pictures. The first picture would be the balance cubes task, which there is a diamond with a colored square that the participant or service member can step shift around on the platform to collide the diamond with the square. This can be done statically or with platform motion. And the second balance task that we looked at was shark hunt, in which the same kind of movement is controlled by the service member, and it controls the boat. And so they navigate their way around this virtual environment to collide with a shark, which has the green above it, the green ball above it, and you can increase or decrease the max speed as well as wave heights depending on the clinician's input. And so we wanted to compare these with the SOT FGA total scores as well as the ABC and DHI total scores. And to account for PTSD, we used the total score greater than or equal to 50 on the PCLM as probable PTSD, and anyone below that was designated not probable. So we looked at the t-test to see if there was group differences as well as correlations to investigate those relationships. Next slide, please. So first, the table 2, we have the correlations, and we see a significant correlation, negative correlations between balance cube statics, that is without platform motion performance, so time to complete, with the ABC, SOT, and FGA. We saw a positive correlation with the DHI score, meaning the higher impact of dizziness that they indicated, or severity of their dizziness on the DHI, was correlated with longer time to complete this acclimation task. With the balance cubes platform motion, we saw negative correlations with SOT and FGA, but neither subjective assessment was significantly correlated. With the shark hunt standard and shark hunt recall, we did not see any correlations between the immersive balance performance on either virtual environment. So to look at how probable PTSD affected performance, we looked at just differences in each of the conventional measures as well as the virtual environment performance. And we saw lower scores on the ABC, so those with probable PTSD had significantly lower scores on the ABC, and they took longer to complete both the balance cube static and platform motion tasks. But we did not see any significant difference between these groups in the shark hunt or shark hunt recall tasks, or the DHI, SOT, or FGA. Next slide, please. So here we looked at just normative values for the ABC, FGA, SOT, and looked between anyone below, for the ABC, 80%, anyone below 28 in the total score of the FGA, and anyone below 80 on the SOT, and we saw differences in these groups on the balance cube static and platform motion for the SOT. So those that had above that normative value did significantly better on the immersive balance task of balance cubes. For the FGA, we only saw this with the balance cube static along with the normative ABC. The only conventional measure that we saw a difference between those groups and the normative values on the Shark Hunt was with the Shark Hunt standard score. And we also saw that the PCLM, the normative SOT, they had significantly lower PCLM total scores, but the PTSD probable group did not see a significant difference in SOT, so we thought that was kind of interesting. So we saw service members with better overall balance were faster on the balance cubes task, but we did not see that stay true for the Shark Hunt standard. There was increased visual stimulation and dynamic platform motivation, so there was a trend which we expected, but we did not see anything of significance. So service members with probable PTSD took longer on both of these tasks, and these relationships demonstrate that there can be utility in immersive balance tasks in the Karen as an adjunct to clinical therapy in this population for assessment as well as continuing on. These are also less time consuming and potentially more engaging than some of these conventional tasks. And our future work will determine if these virtual environments can be pared down to a portable head-mounted display and used in a more traditional clinic setting.
Hi, my name is Mihai Popescu. I am a biomedical engineer and I work in the MEG lab at the NICoE. The poster I am presenting describes one of our recent MEG studies which characterized the brain activity for patients with PTSD during a visual recognition task. This study was conducted under the core MEG research protocol, and Dr. John Hughes is the PI on the study. One of the core symptoms of PTSD is the presence of intrusive memories of the traumatic event, which can be triggered sometimes by sensory stimuli that share some similarities with the stimuli experienced around the time of the traumatic event. Previous studies have proposed that a difficulty with the processing of discriminative information may contribute to the overgeneralization of such trauma reminders in PTSD. Using a visual recognition task, we can actually characterize the brain activity that underlies this processing of discriminative information, which was one reason we used this cognitive paradigm in our study. The participants in the study were 40 NICoE patients. The PTSD symptom severity was assessed with the PCL-5, and the scores ranged between 2 and 71 across our sample of participants. We did not find significant correlations between these PCL-5 scores and age, education, or IQ scores in this sample of participants. Next slide, please. The study had two sessions. In the first session, a series of 86 images of outdoor scenes was shown to the participants, who were instructed to study those images for an upcoming recognition test. Each image was shown on a screen in front of the patient for 3 seconds. In the second session, those original images were shown intermixed with an equal number of novel images while the participants performed the recognition task. All the results described in this study, they were obtained from the MEG data recorded in the second session of the study, the recognition session. The figure on the right shows the main steps of the MEG data analysis. We processed the MEG data and the MRI images from each participant to estimate the evoked cortical currents at a dense set of cortical locations, and then we integrated the power of the estimated currents in 84 cortical regions of a brain atlas and over 100 milliseconds long temporal intervals following the onset of the images. This regional power was then used for statistical testing. Next slide, please. A main result from the analysis of the behavioral data was that PCL-5 scores were negatively correlated with the number of correct responses to original images, which indicates that patients with higher PTSD symptom severity, they recognize the smaller number of original images. In a first analysis of the MEG data, we compared the brain responses evoked by original and novel images across all the participants in this study. This contrast revealed the higher power of the response evoked by original images in a set of brain regions that are shown with red color in this figure, and they included occipital, parietal, and inferior temporal and medial temporal regions that are known to be involved in visual processing, as well as the left inferior frontal gyrus and the bilateral anterior cingulate. Some of these differences were seen very early, starting from 50 milliseconds after the onset of the images, which is consistent with the perceptual priming effect. Priming is considered mainly a pre-attentive process that does not involve any kind of effortful elaboration on the different features of the images. Next slide, please. In a second analysis of the MEG data, we tested the correlations between the PCL5 scores and the regional power of the responses evoked by the original images. The results indicated that higher PCL5 scores were associated with lower power of the evoked responses over frontal, lateral, temporal, and parietal brain regions that are shown in the upper figure, and they are also summarized in the table on the right side of the slide. There are two aspects that I want to mention about these regions. First of all, these effects, they start a little later than the priming effects, 50 milliseconds later, and last for a longer time. Second, this network overlaps what is known as the frontoparietal attention network of the brain, which is consistent with the fact that during the analysis of the complex visual images, attention needs to be redistributed across the different features of the images in the search for those discriminative features that would help to categorize those images as original or novel. Next slide, please. The main conclusions of our study are that participants with high PTSD symptom severity showed a lower performance on this visual recognition task. This lower performance was also associated with a lower evoked brain activity in a cortical network that likely supports the recognition through mechanisms of selective attention to discriminative sensory information. And finally, our findings provide support to recent proposals for testing the effectiveness of sensory discrimination and attention training as part of the cognitive treatment in PTSD.
I'm here. So thank you. So again, I'm going to present two posters. The first one is from the 15-year study. This is the we use the diffusion tensor images to evaluate the longitudinal white matter changes over time. Participants include 285 TBIs and 88 controls. Thanks. All the subjects were scanned at least twice and up to 5 scans. And at average intervals of the after baseline about 6 months, 18 months, 32 months, and up to 15 months. So, and then we grouped the TBI into the total participant into 3 groups, including the mild TBI, non-TBI controls, and the moderate severe TBIs lumped together as one group. How to go to the next one? Okay. So this is showing the color maps of the segment white matter tracks, and the bottom line is 3 figures show the result. So we apply the linear mixed modeling to evaluate the FA, the indicators of white matter change. Look at each average at baseline and FA trajectory after over the times. So the first one we found about 74% of tracks. The moderate severe group, they have the low FA than the mild TBIs and controls. So the figure here is the red colors is the mild TBI groups. Blue color is the non-TBI controls. The green colors are the moderate severe TBIs. So as you can see, for instance, these the first figures, left bottom, cortical spinal tracts. The moderate severe groups has low FA than the mild TBIs and non-TBI controls. And over the time, this difference persisted over the time. And also the mild TBI, about 28% of the tracks, mild TBI groups have higher FA values than the controls at the baseline. But this difference, there's no significant difference in the follow-up scans. So the similar findings for instance, on the right, on the top, cortical spinal tracts. For the trajectories, we found about 18% of the tracks in the mild TBIs have a low FA trajectory between the baseline and follow-up scans. And this trajectory is the difference. They didn't show significant difference in the follow-up scans. And then for the moderate severe TBI groups, the trajectory just persisted. There's no significant change over the times. So the conclusions. Next slide, please. So we found the moderate severe TBI groups has more severe white matter change than the mild TBIs and the non-TBI controls. And there's no evidence of recovery over the times. In the mild TBIs, we found in the baseline they have higher FAs. That could be due to the periregional gliosis that will cause the increase in FA. But this change didn't show up significance over the times that are indicating the recovery. I'm going to present it next. All right, the second presentation, second poster. I will evaluate the meningeal lymphatic factors. So there's a tight, in recent animal studies, there's a tight relationship between the lymphatic systems and meningeal lymphatic factors in TBIs. So we use the post-contrast FLAIR images, T2-weighted FLAIR images to evaluate the meningeal vessel enhancement patterns in the TBI patients. So this study including total 125 TBI subjects and also 52 controls. Next slide, please. So this, after the co-registrations of the pre-contrast and post-contrast and the registrations, we do the subtractions and we do the thresholding and certain values to show more than 50% increase. To show examples here is one of the moderate TBI showing the lymphatic vessels, showing up is a color-coded tiny one. Hopefully you can see it. Next to the superior sagittal sinus. The next one, the bottom one at the cribriform sinus levels. Also, next one, middle meningeal arteries, and adjacent to the choroid plexus in the lateral ventricles. Next slide, please. Another moderate TBI for similar findings, lymphatic vessels at the labels of superior sagittal sinus, and next to the choroid plexus. You can also find on the bottom right, there's a meningeal enhancement of the subject. Next slide, please. So we compare the group difference. We find the the intensity increase for the TBI groups is significantly higher than the non-TBI groups. It is shown here is just 1D plot, on the right 2D plot. Right. So these findings indicate there's maybe impairments of the lymphatic drainage after brain injuries. And currently we are looking at the parenchymal change, which is the lymphatic systems. So hopefully we'll show you the result in the near future. Thank you.
I'm Wei Liu. I'm a physicist at NICoE. Also performing data analysis on perfusion and susceptibility images. First, I want to apologize that I only have this overall slide as I got messed up with the template, so bear with me. This work is try to access cerebral perfusion using dynamic suspended contrast imaging in TBI patients of different severities. Cerebral blood flow, CBF, is critically important for the brain function and to supply the brain with necessary oxygen and substrates. In the literature, CBF patterns seen in TBI patients vary widely. Among these sources of variability in these perfusion studies, severity of the TBI injury and the presence and absence of the clinical symptoms seem to be the most appropriate candidate for further inquiry. This is exactly the object of this study. This retrospective study included a total of 114 male subjects, as shown in Table 1. We have 35 controls, 14 severe, 32 moderate, and 33 mild TBI patients. All the severe and moderate patients scanned under our protocol were included in this study after QA of the DSC images. Since we have a large number of mild TBI subjects, the mild group included in this study were all scanned within 250 days after injury to balance the group size. In Table 1, you can see there's no statistical difference in terms of age, NPSI score, PCLC score among these four groups. DSC images were acquired with an injection of 20 ml gadolinium DTPA. In the paper, relative CBF, rCBF, was normalized to the CBF of the white matter. Table 2 illustrates the rCBF of TBI patients versus the control subject. As we can see, severe TBI patients demonstrate significantly reduced rCBF in bilateral cerebellum, right hippocampus, right brainstem, and also the moderate patient didn't show any altered rCBF in any of these subcortical regions. Interestingly, mild. So having a closer look, this increased amygdala rCBF has demonstrated a negative correlation with the time since the injury, as shown in Figure 1 by this scatter plot. And in addition, this increased rCBF in amygdala in this mild TBI patient also demonstrated a correlation with the headache impact score HIT6. In conclusion, rCBF of TBI subjects with different severities demonstrate distinct characteristics. Severe TBI patients demonstrate reduced rCBF in multiple subcortical brain regions. Moderate TBI patients didn't show any changes in rCBF in any of the subcortical regions studied. Mild TBI patients scanned within 250 days develops increased rCBF in bilateral amygdala. The amygdala rCBF in mild patients demonstrates a negative correlation with the time since injury, which may be indicative of a recovering process from the mild TBI. The increased rCBF in amygdala also demonstrates a linear correlation with the HIT6 score, suggesting activation of the amygdala may play a role in the headache formation. Thank you.
Good afternoon, everyone. My name is Megan Paxton-Willing, and I'm a medical psychology doctoral candidate at the Uniformed Services University and a research extern here at NICoE. Today, I'll be sharing the results from 2 of our sleep studies utilizing data from the clinical database protocol at NICoE. PTSD has been labeled one of the signature wounds of the war on terrorism, with about 12 to 20% of returning Iraq and Afghanistan veterans experiencing PTSD. PTSD is associated with a number of negative effects, including increased alcohol use, increased suicidal ideation, strains on relationship, and decreased quality of life. Notably, these sleep disturbances are incredibly common in patients with PTSD. Some studies actually estimate more than 90% of patients with PTSD experience some form of sleep disturbance. And in fact, these sleep disturbances are so common that the associated nocturnal behaviors can be used to identify patients with PTSD. The existing literature has found a strong correlation between our subjective reports of sleep and post-traumatic stress disorder, but few studies have examined the objective measures of sleep. Further, it's really important that we examine sleep-related factors in active duty populations due to the high rates of sleep disturbances in the military, both before and after deployment, as well as the military-specific factors that can contribute to sleep disturbances, such as sleeping in barracks, sleeping in a deployed environment, or even being stationed on a submarine, ship, or aircrew, which are associated with greater sleep disturbances. So we examined subjective and objective sleep features to understand the relationship with PTSD in service members with a history of combat or mission-related trauma. Next slide, please. Using data from the NICoE intensive outpatient program, we examined the relationship of sleep and PTSD. A diagnosis of PTSD is not available, so post-traumatic stress symptoms were measured using a modified PTSD checklist, with us excluding the sleep-related items to prevent an artificial inflation of the relationship. Patients had an average score of approximately 48 on the PCL, and our self-report measures of sleep included the Pittsburgh Sleep Quality Index and the Epworth Sleepiness Scale, and the objective measures of sleep were obtained via a sleep study conducted during the first week of treatment. Next slide, please. Post-traumatic stress symptoms were related to all subjective measures. Worse sleep quality, greater sleepiness, and greater reports of general and trauma-related bad dreams and nightmares were all positively associated with post-traumatic stress symptoms. However, feelings that sleep was more restorative was related to less post-traumatic stress symptoms. Additionally, we found several objective measures of sleep were associated with greater post-traumatic stress symptoms, including rapid eye movement, sleep latency, time spent in slow wave sleep, arousal index, and all measures of heart rate, including minimum, maximum, and mean. For the objective measures of time spent in REM sleep, wake after sleep onset, and total sleep time, these were associated with lower post-traumatic stress symptoms. Next slide, please. Consistent with the literature, our subjective reports of sleep disturbance were associated with increased post-traumatic stress symptoms. This research additionally provided support for a relationship of several objective measures of sleep and post-traumatic stress symptoms, which may help us to understand this complex and common comorbidity. These findings also provide support for the importance of thorough sleep evaluations for patients with PTSD, ideally including both subjective and objective measures. And additionally, treating co-occurring sleep disturbances may provide an avenue for treatment for patients experiencing symptoms of post-traumatic stress. Can you go on to the next presentation? Thanks. Suicide is an important public health concern globally in the United States and particularly within our U.S. military. A number of factors have been implicated with sleep hypothesized to increase risk of suicidal ideation. Some sleep disorders, including sleep apnea and insomnia, have been found to be associated with increased suicidal ideation and number of attempts. Given the high prevalence of sleep disturbances in the U.S. military, it's really important for us to understand the role of sleep on suicidal ideation specific to our service members. Subjective sleep reports, including insomnia symptoms and nightmares, have been found to be positively related to suicidal ideation. But presently, the research has really focused on these subjective measures of sleep, with very few studies examining the objective measures of sleep factors. Notably, the studies that have utilized objective measures have had inconsistent findings, with some identifying respiratory disturbance index, apnea-hypopnea index, and oxygen desaturation index being positively associated with suicidal ideation, but other studies not finding these same relationships and instead finding time of sleep onset predictive of suicidal ideation. Therefore, we tried to examine the sleep-related factors, both objective and subjective, in relation to suicidal ideation in service members. Next slide, please. Like our previous study, this study used clinical data collected as part of the NICoE intensive outpatient program. Our sleep measures included objective ones collected as part of the overnight sleep study. Sleep quality is measured by the Pittsburgh Sleep Quality Index, the Epworth Sleepiness Scale, and general bad dreams were measured via the PSCI, and trauma-related bad dreams and nightmares were measured using the PCL. We measured suicidal ideation using the patient health questionnaire and made this a binary variable to enable our logistic regression. Next slide, please. REM sleep latency was associated with increased odds of suicidal ideation, and time and stage N2 sleep was associated with decreased odds of suicidal ideation. Notably, all of our subjective reports of sleep were associated with increased odds of suicidal ideation, with trauma-related bad dreams and nightmares being associated with an increased odds of 9.90 for suicidal ideation. Please note that the analyses for both the ones using the Pittsburgh Sleep Quality Index and the PCL for bad dreams and nightmares controlled for the alpha-1 blocker prazosin, because this is a commonly prescribed treatment for trauma-related nightmares. And additionally, these items were both categorical, so the results displayed here in the table are for the most severe category. Next slide, please. The present results add to the existing body of research examining objectively measured sleep quality as it relates to suicidal ideation. To our knowledge, this was the first study to find REM latency and time spent in stage N2 to be significantly related to suicidal ideation. However, these relations were small, suggesting these factors may not play a meaningful role in suicidal ideation. Further, there was no evidence of a relationship between objectively measured sleep indices previously identified in the literature as suicide correlates. Consistent with the existing literature, however, all subjective sleep reports in the present study were significantly associated with suicidal ideation. These measures also were the only ones to produce meaningful odds ratios in our findings. We were particularly excited by the findings of the large increase in odds of suicidal ideation for patients experiencing severe trauma-related bad dreams or nightmares. While these results show promise in the relationship of suicidal ideation, additional research is needed to examine this in additional samples. As suicide is a current public health crisis facing the U.S. military and nation as a whole, it's important that providers consider all possible avenues to mitigate suicidal risk. Therefore, as an adjunct to a provider's standard procedures when conducting a risk assessment, they should evaluate subjective sleep symptoms, particularly bad dreams and nightmares, in combination with comorbid conditions. Though requiring replication, our present results suggest bad dreams and nightmares, particularly the trauma-related ones, hold promise as a possible point of intervention to help reduce suicidal ideation in active duty service members. Thank you.
Good afternoon, everybody. My name is Catherine Rizzuto, and I am a clinical research assistant at the Center for Rehabilitation Sciences Research. Today, I will be walking you through the Transcranial Magnetic Stimulation, or TMS program, that we have going on at the CRSR. We have 4 studies that use repetitive TMS, or rTMS, as a treatment intervention focusing on patients who have mild traumatic brain injury, post-traumatic stress disorder, early onset of Alzheimer's disease, chronic regional pain syndrome, as well as chronic neck pain. Can you go to the next slide, please? So what exactly is TMS? TMS is a non-invasive, low-risk neuromodulating rehabilitative method that uses magnetic fields to aid in the strengthening of weakened neural connections and reinforce neuroplasticity through the stimulation of nerve cells within the cerebral cortex. rTMS has shown immediate and favorable outcomes in patients with varying neurodegenerative and cognitive impairments, as well as psychiatric disorders through the alleviation of associated symptomology that normally require intensive rehabilitation. MTBI, PTSD, EOAD, CRPS, and chronic neck pain are common conditions among servicemen and women. Existing therapies for these show limited efficacy, are often costly to the patient, can have dangerous side effects, and sometimes include pharmacological intervention to help alleviate symptomology. The persistent use of pharmacological intervention has led to a dependency on opioids in some service members, which is a prevalent issue among service members seeking a remedy for pain. TMS, however, is safe, tolerable, affordable, and eliminates this reliance on substances as a means for pain reduction. Next slide, please. So due to the extensive nature of the methodology for the 4 studies I'm talking about today, and within the interest of time, I'm only going to be giving a brief overview of the methodology. However, I did want to let it be known that I'm happy to provide further information on the specific methodologies for any of the 4 studies that are going to be talked about today upon request. Just a brief overview of what we do consistent across the 4 studies. A clinician determines the location of the brain regions to be targeted for each therapy, as well as the energy settings. The clinician performs the initial motor threshold, and a provider trained in TMS performs any subsequent sessions. The resting motor threshold is determined by seeking an area of cortical stimulation within the participant. The first ongoing TMS study that we have is a double-blinded, sham-controlled, prospective, and randomized clinical trial where patients presenting with a comorbidity of MTBI and PTSD receive bilateral rTMS to the dorsolateral prefrontal cortex in an effort to alleviate symptomology that's associated with their conditions. This study utilizes both high and low frequency stimulation with 3,500 pulses to the left prefrontal cortex and 15 of 1 Hz stimulation to the right prefrontal cortex. The next study is a case report that assesses the success of rTMS alongside various cognitive training exercises in delaying the early onset Alzheimer's disease symptom onset. This study utilizes high frequency stimulation bilaterally to the dorsolateral prefrontal cortex. Another case study using rTMS sought to alleviate CRPS through unilateral stimulation of the patient's motor cortex, and this study utilized high-frequency stimulation. And the final study within the CRSR portfolio is a progressive, double-blind, randomized, sham-controlled study that's currently within the developmental stages. This research endeavor is aimed at treating chronic neck pain through the employment of personalized rTMS, which we refer to as PRTMS. PRTMS is a variation of TMS that uses patient self-reported outcomes and quantitative electroencephalogram readings for a more personalized form of rehabilitative therapy. We plan to use high-frequency stimulation pulses ranging between 8 and 13 Hertz to be delivered either to the central zone cortex, the frontal zone cortex, Broca's area, frontal precortex, or other local brain areas at the stimulation depth of 2 centimeters. This, as well as the amount of pulses that we plan on delivering, will be dependent on the patient that we have. Next slide, please. So the study utilizing rTMS for PTSD and MTBI is ongoing. We did have to stop efforts due to COVID, but overall tolerability has been high, with the average pain of stimulation reported at 1.84 out of 10. Interim analysis has indicated that compared to participants receiving a sham rTMS, those receiving the active, like their patient, the PTSD symptom and concussive symptom scores improved, although this has not been significantly yet, and our neuroimaging and biomarker analysis has not yet been completed. The case report for early onset Alzheimer's disease data analysis is also still ongoing, and the patient has completed 2 cycles out of the 3 that we plan to complete with them. Overall, the tolerability has been high, with the average reported pain of 2.42 out of 10. The patient scores improved slightly for the cognitive functioning inventories. The cognitive training from baseline showed overall improvement, but specifically memory and attention have shown improvement. Next slide, please. The case report utilizing rTMS for CRPS has been completed. Overall tolerability was high, with the average reported pain of stimulation of 1.51 out of 10. While the patient's daily pain score fluctuated based on the patient's activity level the day before, the overall trend was downward. By the end of the 20 sessions, the patient endorsed both a 30% reduction in total pain in all extremities and greater functional mobility and independence. And the PRTMS, we don't have any results yet. We hope to determine the effectiveness of it as an adjunctive therapy for chronic neck pain. Overall, these studies will guide the expansion of the rehabilitative possibilities of TMS. TMS has shown great promise as an adjunctive therapy for multiple conditions that require intensive rehabilitation as its growing form of therapy within the military health system. Thank you.
Hi, so my name is Diana Nora, and I'm the Clinical Research Coordinator for the TMS study for depressive symptoms associated with concussion at NICoE. So today I'll be going over our preliminary results regarding the safety, feasibility, and tolerability of TMS in our study population. Next slide, please. So as many of you know, depression is a common side effect after a concussion and can be quite debilitating. Although transcranial magnetic stimulation is an approved treatment by the FDA for medication-resistant major depressive disorder, little is known regarding the treatment as an option after a concussion. So our focus of our study was to examine TMS as an effective treatment option for service members experiencing depressive symptoms with a history of concussion. Next slide, please. So for our results, we broke down our findings by safety, feasibility, and tolerability. So for safety, we mainly looked at the daily TMS side effects and related adverse events. For feasibility, we looked at the outcomes of recruited and consented participants. And then for tolerability, we looked at the percentage of fully completed TMS courses by the participant. So for our safety and tolerability analyses, we mainly focused on data from 5 participants that were randomized and completed any number of TMS sessions. So looking at the clinical characteristics of these participants, the baseline depression severity ranged from moderate to severe, with most participants in the moderate range. And all participants had existing chronic pain, headaches, and an anxiety disorder. Next slide, please. So looking at our safety results, this figure is showing the number of TMS sessions with side effects by participant. And then you can see the type of side effects on the right there. So overall, the common side effects were headache and neck pain and stiffness, which is consistent with the TMS literature. We have not had any seizures, serious adverse events, or unanticipated problems to date. And one interesting thing to note is that we did have one participant experience transient phantom limb pain during 2 sessions. And this is something that did occur on a normal basis for them, but since it occurred during the TMS sessions, we don't know if the TMS had any effect on it. So this may indicate that there may be side effects unique to the military population that should be further explored. Next slide, please. So looking at the graph on the left there, we recruited 54 participants from various sources. 30% of the participants did not meet initial inclusion-exclusion criteria, and 24% declined. And the most common reason for decline was time commitment. And then looking at the figure on the right, in total, we consented 10 participants. 4 out of 10 participants were either excluded or withdrawn. So this is slightly higher than our anticipated early dropout rate, but we think that the small sample size may also be a contributing factor. Next slide, please. So this table is showing the percentage of total TMS course completed by participant. So overall, the tolerability of the TMS course was good. We had the course included 20 sessions, and so we had one participant withdraw due to headache and neck pain, but all other participants completed most TMS sessions. But overall, again, a larger sample size may be needed to assess tolerability, especially due to the coexisting conditions. Next slide, please. So the limited preliminary results suggest that TMS is safe, feasible, and tolerable in a military population. There may be a need to reduce time commitment involved in the TMS course due to the unique obligations of service members. And overall, future research and larger samples definitely needed to assess the aspects that may be unique to the military population, such as different side effects or coexisting conditions. Thank you.
Hi, I am Dr. Allison Winters-Fisher, and I'm a wellness coordinator at the NICoE. I am a dance movement therapist and a yoga instructor, and I have a PhD in mind-body medicine, which I recently completed at Saybrook University. This poster is a representation of my recently completed dissertation. It's entitled Embodied Warrior Wellness, Dance Movement Therapy-Based Mind-Body Medicine in an Integrative Medicine Treatment Program for Military Members with Traumatic Brain Injury. It was a holistic case study of NICoE's integrative IOP treatment program, 4-week program. And the case study really looked at the program from 3 major perspectives. One is the program is executed, the program is experienced by the patient, and then the program is experienced by the researcher practitioner, which is me. I drew from several sources of data, both qualitative and quantitative, field observations, public archival data such as annual reports, the NICoE website, mission statements. I also pulled from several IOP wellness surveys. The patients fill those out in weeks 1 and 3 of the 4-week program, and there was quite a bit of qualitative and quantitative data available in those surveys. And then I also pulled from embodied data. There were several dance and art interpretations done throughout the process, and you'll actually see a little bit of that in the slide. Analysis. There was qualitative and quantitative analysis completed. There was on the qualitative side, there was a thematic analysis, mostly of the data from the wellness surveys. There was a lot, a lot of data. And then also an embodied art-based approach was used. And you can see some of the products of that on the slide. So there's an image in the lower left-hand corner, and that is from some of the movement interpretation. And then there's also some hand-drawn images, and that was from some of the art-based analysis. And then you can see in the results box there some themes and patterns that came from the thematic analysis. So particularly for the patients, some shifts in self-awareness, big themes around transition, emotional struggle was a big focus, moving from discomfort to comfort, so just to name a few. And then below that, you can see there's that's some of the descriptive statistics. One of the questions, or two of the questions that are asked on the survey, ask the patients to rate their own level of mind-body awareness, and then also their confidence with mind-body practices. And you can see from week 1 to week 4, there was an increase, 48.3% for the mind-body awareness and 51.6% for the confidence levels. So out of all of that, there were some main discussion points that came up. One is just there seems to be a relationship between interoceptive awareness and leadership. So interoceptive awareness being that sense of your kind of inner landscape, inner sensations. Also, the culture of wellness there seems to be a relationship there between a collective understanding in the community of what integrative medicine even is. You know, it became clear that people may have different ideas and thoughts about what that means. Provider burnout was something that came up too, perhaps an opportunity for mind-body skill building, and post dissertation, I've seen this relationship in the work I've been doing with the resiliency offerings since COVID started. So interesting to see that manifesting outside of the dissertation as well. And then question about whether introducing these mind-body skills earlier in people's military careers might help with things like resilience and readiness. And then lastly, professional standardization of IM practitioners might be helpful to meet military and veteran needs within the military healthcare system. So that is it in a nutshell, and I am open to questions, so feel free to put things in the Q&A box if you'd like. Thanks.
Hello, my name is JD Hogan. I'm a statistician and music therapist working with Creative Forces Network as their senior informatics specialist in support of Henry Jackson Foundation and National Endowment of the Arts. I'm under Dr. Caban's informatics team. And today I want to present some initial evaluation of music therapy and standardization within Creative Forces. And one of the main challenges we have within the network is we have 12 different sites with 3 different types of disciplines for therapy, and even within music therapy alone, we have 11 different therapists. So standardization between all of the different therapists and within the sites of needs of the sites is pretty challenging. But we try to do this in 2 different ways. One is the workload reports, where the therapists can capture their productivity, like the number of sessions or hours they spend doing sessions or time spent documenting. The second is we built templated notes, and the music therapist spearheaded this initiative within our network. And we built the notes using a SOAP note format, and we put it in TBI portal so that the different sites could access it and we could look at the data. There are some key concepts that I want you to take away from this presentation, and it's that it looks like standardization is possible within this network, even with all of the challenges. But we did this through making a standardized format, but creating pivot points so that the therapist could still provide individualized patient care. But it looks like the templates might be allowing for additional benefits. And one of those is just giving us access to research in the future, so that we have that structure in place. But another is potentially a decrease in time documenting. So this chart we have on this slide, it's comparing music therapy, which is a discipline with standardized note templates, versus art therapy and dance movement therapy, where there's not really standardized note templates. And you can see that music therapy has lower time documenting because they spearheaded this, but there might be other factors in play. So the big takeaway from this is it looks like it's working, but we need to look further into it. Next slide, please. But we have a therapy protocol where we assess the patient at the beginning of treatment. We set goals and interventions, and we reevaluate as we go. And when we discharge, we offer community engagement. Next slide, please. And this slide is showing how goals change across sessions. So you can see that goals really do change across sessions, and that we are reevaluating and seeing what does the patient need. I wasn't able to put this on the slide, but when we discharge, it's often consistent across sessions. And when we do discharge, we are offering community engagement on top of that. Next slide, please. For interventions, we are changing our interventions based on the changing goals as well. And you can see that assessment interventions were 41% of the interventions in session 1. That dropped down to 1% in session 2. So you can see that we are doing assessments in the first session. We are establishing and reevaluating goals and interventions as we go. We are discharging and offering community engagement. And Nate McLaughlin is our music therapist here at NICoE, and he wanted me to read something at the end of this based on his experience with the notes. So, and I quote, "I think the key benefits of these note templates for me as a clinician are that they keep the type of information I pay attention to and record consistent between individual sessions, and also allow me to capture the unique details of each clinical picture across individual patients. These are really critical concepts for tracking progress through a process as fluid and dynamic as therapy, perhaps even more so among the creative arts therapies. I can take better notes and in turn take better care of my patients while also doing so more efficiently. And so the key takeaway from this is it looks like standardization is working and it looks like it's helping. So thank you.
Okay, thank you. So I am Liz Freeman. I'm the lead dance movement therapist for the Henry M. Jackson Foundation for the Advancement of Military Medicine in support of creative forces, and I conduct my work here at the NICoE. So I'm going to speak a little bit more broadly about some of the standardization efforts that JD just brought up for us. This poster was co-authored by Rebecca Vaudrey, Melissa Walker, J.D. Hogue, and Donna Betts. Creative Forces is a partnership between the National Endowment for the Arts, the Department of Defense and Veterans Affairs, and state and local arts agencies. The goal of Creative Forces is to improve the health, wellness, and quality of life for military and veteran populations exposed to trauma, as well as their families and caregivers. So this poster demonstrates the standardization of clinical operations for creative arts therapies, and this was aimed to support and drive clinical research across the Creative Forces network. These efforts to standardize have examined our clinical interventions, assessment and treatment protocols, our documentation procedures, and program evaluation and research. The 4 areas of focus followed the 6-stage standardization process you see on the screen, and those were aligned with the pillars of Creative Forces, which are clinical, capacity, and community-based work. For capacity, we looked at how this process informed our conceptual frameworks, which led us to strategic and intentional research questions across the disciplines currently involved with Creative Forces, which are art therapy, dance movement therapy, and music therapy. Go to the next slide, please. So these efforts to standardize across these 4 areas of focus resulted in the following outcomes. For clinical practice and intervention, we developed treatment manuals and training videos. And you can see to the right figure 2, which depicts one of the training videos that was created to inform clinicians of interventions and to train them to the standards, which was one of those previously mentioned procedures. This standardization of practice, so not only ensuring that our clinicians are employing the same interventions, but that they are actually administering them in a similar manner. This has been a critical step forward for us as a network to be able to generate strong multi-site research in the future. For assessment, we have developed and implemented standardized assessment protocols. For documentation procedures, as JD was mentioning, we have developed these standardized note templates across all of the disciplines for both individual and group sessions. And you can see there, figure 3, an example of what one of the standardized music therapy note templates looks like as it is embedded into the TBI portal. For program evaluation, we have developed logic models and conceptual frameworks for practice and research, and program evaluation has of course been bolstered by the standardization of documentation as well. Outcomes and observations from this process has allowed clinicians to identify gaps in knowledge and to identify future areas for research study. Go to the last slide, please. So figure 4 here represents our efforts to look at common symptoms and treatment goals across the disciplines so that we're better able to understand where there's some overlap in clinical practice and where we can really build on those individual strengths of the disciplines. This allows us to more specifically target treatment outcomes and to better identify which interventions are most useful for particular symptoms or goals or subpopulations. This is in line with that revise and refine procedure, allowing us to identify what the creative arts therapies are both uniquely and collectively addressing to inform that research agenda. This has also been highly valuable for us as practicing clinicians because the more refined we are able to get with what goals and symptoms we are treating across the disciplines, the better referrals we are then able to get from other providers. This specificity also helps us develop even more effective assessment protocols and documentation procedures, which of course allows for stronger research. So in conclusion, standardization has helped us maximize efficiency in clinical practice, community engagement, and capacity building throughout the Creative Forces Network. Thank you.
Hello, my name is Hannah O'Malley, and I'm coordinating our study evaluation of quantitative EEG during art therapy in service members with comorbid post-traumatic stress and traumatic brain injury. And I'm doing this in collaboration with our principal investigators, Dr. Chandler Rhodes, Melissa Walker, and our art therapist, Adrian Stamper. Then Jasmine, next slide, please, and thank you for organizing this. So art therapy is a distinct profession that can enrich people's lives through active art making and engagement in the creative process in a psychotherapeutic relationship. Quantitative electroencephalography, or QEEG for short, measures the electrical activity of the brain through sensors that are embedded in a mesh cap that's worn over one's head, similar to a swim cap. At the NICoE, we found that art therapy anecdotally is an effective treatment for post-traumatic stress symptoms in service members, but there's still limited research to support this. There's also been preliminary work using fMRI, functional near-infrared spectroscopy, and electroencephalography, but there's yet to be a study using QEEG during art therapy. In collaboration with the National Endowment for the Arts Creative Forces, the goal of this longitudinal study using mobile QEEG directly before, during, and after art therapy is to bring us closer to an understanding of what neurobiological changes may be taking place when service members engage in art therapy. Next slide, please, Jasmine. So for this study, we'll be working with active duty service members with comorbid post-traumatic stress and mild traumatic brain injury with previous combat exposure and no previous art therapy exposure. The study design entails a baseline assessment and post-therapy follow-up where we'll ensure eligibility and measure pre- and post changes in depression, post-traumatic stress, somatic, anxiety, and alexithymia symptoms. And we'll also account for resilience and identity measures. Service members will do 6 art therapy sessions while wearing the QEEG cap so that we can measure their resting state EEG pre- and post-art therapy and throughout the art therapy session. Next slide, please, Jasmine. So with the collected data, we'll compare QEEG measures such as regional and global power and coherence with their self-reported symptoms and individual traits and life experiences. In the future, this work can help us understand the impact of art therapy on brain activity. It can also help to create personalized art therapy for service members with complex comorbidities. And finally, mobile QEEG is a promising research tool for future research protocol. So thank you very much, and we look forward to sharing the results of the study with you at next year's research fair.
Thank you, Amy, who's adjusting the camera so that I can be seen. Hope everyone's doing okay out there. I wanted to say a couple of things before we transition to the third piece of the fair. I think the first thing that I'd like to say is that is an acknowledgement that the presenters represent just a range of different disciplines. And different levels of professional development, and for what it's worth, I think they did a really nice job today, both with the presentations, the format of this, and I just want to express a bit of gratitude there for the efforts and the quality of the presentations. Additional thanks I wanted to offer to NICoE leadership, Captain Greenhalgh, who is recently retired, director here for a number of years. Dr. French, Mr. Eddie Bueno, very supportive of the concept of the FLAIR, and so, I'm seeing a camera fall over. Okay, we're back. Without top-level support, really at NICoE, these things wouldn't be possible. And I honestly look forward to seeing what the next iteration of this might look like, up to and including involvement of some of the other Intrepid Spirit sites. Also, a quick acknowledgement to the FLAIR planning team here, which I think did a great job over a number of months, and I'll mention just some names quickly. Jasmine Moxley, who's moderating, Travis Dittmer, Teresa Wu, and Paul Frimpong for a number of weeks and months really to help prepare for this. So thank you to those individuals. The final piece of our FLAIR today will be a presentation by Ms. Sarah Kruger. Ms. Kruger's presentation provides an overview of the NICoE Computer-Assisted Rehabilitation Environment, or CAREN as an acronym, and really how that has evolved over the past 10 years. I would note that Ms. Kruger has been here at the NICoE since the cutting of the ribbon, and so she has a historical perspective, both on the CAREN and I think about the NICoE, when it started, how it started, and how it's developed. So she'll talk a bit about the clinical and research utilization of the space in the CAREN and how that's led to collaborative innovation. Ms. Kruger is a biomedical engineer in charge of the operation and management of the CAREN laboratory. Primary responsibilities include running the CAREN for clinical care, collaborating with clinicians and scientists to develop research protocols for grant funding and IRB submission, and creating new virtual environments to support clinical and research activities. She's an investigator on multiple research studies to seek to utilize virtual and augmented reality-based technologies in innovative ways to advance understanding of service members with TBI and associated conditions. She had asked me to do a much shorter introduction, and she hopefully will indulge me with a bit of a longer bio. But please welcome Ms. Sarah Kruger.
Did he thank Amy? Amy's been fixing everybody's angles here today.
Oh, you gotta get your angle.
I know. I should have put lipstick on. It would have been the first time in 6 months I could have put it on. So as mentioned, I'm the biomedical engineer who manages the computer-assisted rehabilitation environment laboratory here at the NICoE. And I was hired in 2010 before the ribbon was cut, so I do know where all the bodies are buried. That being said, I'm going to give the disclaimer and say that I also have no financial disclosures of conflicts of interest. The intent of this presentation is to provide an overview. I'm going to move this little guy. Oh, thank you. Sorry, I was covering my notes. The intent of this presentation is to provide an overview of the NICoE CAREN laboratory and describe how over the last 10 years, the clinical and research utilization of the space has evolved through collaborative innovation. We've leveraged insights gained through clinical care to formulate relevant research questions. Then, through cooperative relationships and scientific partnerships, we've sought to expand the system's capabilities in an effort to improve outcomes for our service members. Maybe. And the engineer is having technical issues, but I'm going to figure it out here in a second. Maybe. Or, there we go. So there are currently 4 CAREN systems in the DoD. We are lucky enough at Walter Reed to have 2 on our campus. I previously ran the CAREN laboratory at Old Walter Reed before coming to the NICoE, and I've been working with this system for over 13 years now. For those who are not familiar, the CAREN is a large virtual reality-based system that combines multiplanar motion with immersive virtual environments projected onto a curved panoramic screen. The motion platform can move up, down, forward, backward, right and left. Oh, maybe. Up there it goes. And can rotate in all directions. There's an embedded treadmill on the top surface, and that can go over 11 miles an hour. So this is a safe, controlled place to sometimes go from walking to jogging to running to sprinting. Everyone goes in a harness. The embedded treadmill works in combination with a motion capture system that tracks movement in real time via reflective markers. This allows for movements on the platform to either control the progression of or allow for interaction with the different virtual environments that are out on the screen. So this graph illustrates our utilization over the years. Both clinical care and research are represented. The early years were heavily clinical. However, as the clinical template at the NICoE changed, a lot of that was adding alternative modalities, which you've heard about in the previous talks. There became an opportunity to start rolling in some research protocols. So as you can see here, we've had more than 1,000 clinical sessions, and when we look at the mix of things, the utilization of the space has been about 66% clinical and 34% research. I'll go into that here in a little bit. For clinical utilization, as I said earlier, the Cairn provides a safe, controlled setting for care. It's another tool in our clinicians' arsenal, and it's used as an adjunct to standard of care modalities. Most clinical referrals come from physical and occupational therapy, and while the system primarily serves NICoE IOP patients because of its physical location, I've also seen referrals from Old National Naval and now Walter Reed, and we also have some outpatient services coming from Fort Belvoir. The NICoE IOP patients, because they're only on site for 4 weeks, may come in once or twice if they are referred down, whereas outpatients may be seen multiple times over a course of weeks and in some cases months. Regardless, before going out on the system, all patients receive an overview of the technology as well as the system's safety features. I'm an engineer, I'm a stickler for that safety. Then they are run through a series of preliminary virtual environments that I developed at Old Walter Reed to help acclimate service members to the system so that they become comfortable moving in the harness and on the platform, as well as adjusting to the visuals on the screen, which does wrap past their periphery when they're in the center of the space. After completing the preliminary virtual environments, the clinician can choose from a variety of different options in order to tease out symptoms, work on compensation strategies, determine tolerances, or introduce challenges beyond what is currently capable in the conventional space. Okay, I'm going to see if I can get these movies moving. There we go. So when I started at Old Walter Reed, there were only 3 virtual environments. So to get buy-in from the clinicians and the patients, who at that location were primarily individuals with amputations, I built a lot of new virtual environments that they wanted to work on skills that they felt were important. Those environments came with me when I arrived at the NICoE, where I had new clinicians and new brains to tap into. We were able to build new novel environments and expand the existing environments even further by making changes specifically for those with traumatic brain injury. We added a lot of different layers and new settings to include audio and visual stimuli, cognitive tasks, as well as background options and additional platform motion settings. The environments can essentially be as busy or as bland, as easy or as challenging as the clinician desires or patient warrants. Through our clinical utilization of the system, questions arose. Some we knew that we could look at retrospectively, but others needed to be tackled prospectively. When developing research questions, we tried to leverage things we'd seen clinically, not just on the Cairn, but within the NICoE and here at Walter Reed. We wanted to use the technology to conduct translational research that would improve outcomes for service members with TBI. But as an N of 1, this could not have been possible without help. So I sought out partners who either shared my questions or who had questions that I thought the Cairn could help answer. This brought a number of wonderful clinicians and researchers into my cold, dark space, and with them came new insight. Through shared knowledge and resources, hard work and perseverance, we've created some new virtual environments to investigate some really interesting questions. Our retrospective research has focused on the virtual environments we've been using for care for over a decade. We sought to determine whether performance that we saw on the Cairn platform differed based on the diagnosis of the service members in the space. We also have looked at whether or not Cairn performance and conventional outcomes are correlated, and that's ongoing. You saw Dr. Rosen present some of that earlier. Prospective research started with questions about multi-sensor integration from Dr. Doug Brungart. The study that I'm going to talk about with him actually was a conversation we had back in 2008 in a different cold dark room with a screen that was brick, and it wasn't possible, but here our screen is cloth and sound can travel through it. So He's senior scientist for the Hearing Center of Excellence and is over at the National Military Audiology and Speech Center at Walter Reed. We've progressed to the examination of a novel PTSD intervention with Dr. Mike Roy, who's at Uniformed Services University. And most recently, we've been investigating whether the Karen can actually be pared down due to advances in technology and head-mounted displays that have been possible in the past couple of years. We want to be able to see whether or not we can use both the Karen and a pared-down version to enhance vestibular physical therapy. And that's with Dr. Panada Sessoms and her group out at the Naval Health Research Center in San Diego. Okay. Retrospective research would not be possible at the NICoE without the early efforts of Dr. Tom DeGrava. He put together a clinical research database protocol which started capturing the clinical outcomes of service members who are part of the NICoE IOP program. He also obtained funding for the NICoE Network Research Program, which brought a wonderful postdoc into my space who helped facilitate our ability to compile, quality control, and analyze Karen clinical data efficiently. Then through the combined efforts of clinicians and researchers, we've been able to look at the data in a comprehensive fashion. So I'm going to hit on a couple of the applications Dr. Rosen spoke about earlier, but these preliminary virtual environments, they're utilized by all service members when they come into the space and when they first go out in the system. I originally developed them really just so that people would be comfortable, but we've seen some interesting results when we've looked at them retrospectively. So the balance balls application, this was not on Dr. Rosen's slide. This is an acclimation to weight shifting. Platform is static. They've got 2 reflective markers on their back above the pelvis, and they are synced to the movement of a yellow ball on the screen. They are asked to weight shift to hit targets that pop up sequentially at random locations. So again, feet planted, just getting comfortable moving in the harness. We progress to the balance cubes application. It's a similar task, but now we're allowing them to step shift. We're actually encouraging them to move more on the platform. Patients are synced to the white diamond with the same marker placement as described for the balance balls. They must step shift to hit colored cubes that pop up. They appear one at a time at random locations. Steps to the right or left, move the diamond accordingly on the screen. Steps forward of center, move the diamond up, and steps rear of center, move the diamond down. The main outcome measure for these is just time to complete the task. That being said, the clinical provider in the space is looking at quality of motion. What is the strategy? How are they kind of attacking this scenario? And maybe it wants to stop that before it'll let me go. There we go. So as I said, I originally developed these applications so that people would be comfortable moving on the platform. I did not anticipate that the outcomes would be all that important. However, we found that performance on these tasks was significantly poorer in service members with comorbid PTSD when compared to those without. Service members with comorbid PTSD took about 15 seconds longer on the balance balls and about 10 seconds longer on the balance cubes. Now these are done first in my space, so it could be the novelty of the task, could be that they're on a floating metal island, could be that they're in a harness, but it's consistent. We saw that this trend continued when we added some healthy controls to the mix. Time spent on the balance balls significantly predicted participant category between TBI PTSD and control groups. And then for the balance cubes, time spent significantly predicted category between TBI PTSD, TBI no PTSD, and then again between the TBI PTSD and control groups. The Sham-Controlled application, that was also described a little bit by Dr. Rosen. This is a more dynamic virtual environment. The platform is synced to move with the waves that appear visually, and the service members are synced to control the boat. Their objective is to navigate the boat towards target objects, which in this case are sharks. Everybody wanted to hunt something, and this was as PC as I could make it. Steps forward of center, move the boat forward on the screen. Steps rear of center, move the boat in reverse. Weight shifts to the right or left. Bigger the weight shift, the bigger the turn on the screen. Platform also banks with the turns. The clinician can determine how difficult the platform motion is to increase or decrease the challenge. Their objective is to go after the sharks with the big green ball. Each time they hit that target, the green ball jumps to their next target. So it's time to complete, but that's rolled into a composite score, which also takes into account any penalties that are incurred. So if they hit an island or another shark, that is not the target, they get a really good Homer Simpson dough, and they get some points deducted from their total score. We oftentimes go through this a second time. Clinicians wanted to have a recall task added over the top. Service members are not allowed to count on their fingers or count out loud. That really upsets physical therapists. So I'm asking them to count red cubes that pop up to the right and left of the boat. We do a variety of other color cubes too. They're just asked to keep track of the red ones. So same task of hunting the sharks, but just this added recall task on top of it to add a little bit more cognitive. So through an ANOVA, we revealed no statistically significant difference in performance when it was just the shark hunt task when we looked at PTSD status. However, when we looked at performance of shark hunt recall, hopefully that'll work, service members in the no PTSD category performed better than those with subdiagnostics, so that's a PCLM of 31 to 49, and probable PTSD, which is PCLM greater than or equal to 50. In addition, whereas service members in the no PTSD category showed an improvement from the first run shark hunt to shark hunt recall, there was less significant improvement in performance in the PTSD categories, so not a lot of learning going on there. That being said, we didn't have a ton of people in the PTSD only. It would have been nice to look at PTSD only, but our service members here, our population doesn't really allow for that. So then going on to conventional measures, I work primarily with physical and occupational therapists. Vestibular is a huge part of why the physical therapist might refer a patient down to the space. With the platform motion, with the visuals that we can add on the screen, it really allows them to go beyond that space. Some standard applications, assessments that are carried out in physical therapy for vestibular are the functional gait assessment and the sensory organization test. Those are objective measures of functional gait and balance, whereas the ABC and the DHI looking at self-reported balance competence and then self-perceived dizziness. Also had the pleasure of working with Dr. Hyland and Dr. Milkey. We did a quick analysis for MHS a couple of years ago, and that was looking at the WAIS, the DCAFs, and trails A and B. So cognitive function, verbal, nonverbal, executive function, and then with trails, visual attention and task switching. So how do these things compare to some of the performance we see on the platform? So this is going to be familiar if you were here earlier and saw Dr. Rosen's talk. We looked at the objective measures and we looked at clinically relevant cutoff scores for the vestibular outcomes. And what we saw is that the functional gait assessment and the sensory organization test, the objective measures, showed stronger relationships in both the balance cubes and shark hunt. The balance cube task, where the platform was kept flat with no platform motion, actually showed the strongest relationships even with some of the subjective measures. For the neuropsychological outcomes, we found that longer time spent on the balance cubes and balance balls, so those simple weight shifting and step shifting tasks, was associated with slower processing speed and lower executive functioning. We're going to dive a little bit more into neuropsych stuff as we continue our retrospective analysis, but for the time being, I'm going to switch over to some of our prospective research, starting with our collaboration with Dr. Brungart, which at this point we've carried out multiple phases. I'm going to focus on the first phase of the research due to time. With Doug came a whole team. It's a wide variety of disciplines. I am very grateful and appreciative of the parts that each of these individuals played along the way. In combat situations, the detection of targets and evasion of threats may occur at the same time and may occur while our service members are moving. The thinking is that this could be a hidden operational problem for service members with mild TBI. So the objective of this project was to develop a multisensory task where we used audio cues to localize and identify targets while individuals were walking out on the care and treadmill. This tested on healthy controls as well as those with blast-related TBI. The grants for this study allowed us to enhance our infrastructure in the care and space. So this is adding some actual bells and whistles to the bells and whistles we already have. We added a custom 64 speaker array. The Karen at the NICoE is the only one that has enough physical space behind it for a speaker array, and this is why this wasn't able to happen at Old Walter Reed with a brick screen. Our cloth screen allows sound to travel through it and can be heard out on the platform with very little impairment. The location of each physical speaker was mapped to the different virtual environments that I created. So each speaker had a coordinate in the virtual environment, and then we added some additional distractors for the audiovisual task. Essentially filled the whole screen. Some folks were not happy with that one, but it's a good task. The first task we had folks complete was an audio localization task. So we removed those physical speakers. They were hidden. We removed the virtual speakers. They were hidden in the environment. And 5 markers were placed on a headband and synced to a cursor on the screen. Participants were asked to start over the white circle in the center of the screen, and then a pulse noise was chosen from one of those speakers at random. Their job was to then move their cursor to where they thought the sound was playing, and then using a button on a game controller, kind of lock in their position. So we were able to look at the coordinates of where the speaker was versus where their crosshair was to determine their accuracy. I'm going to give you a taste of the sounds, hopefully. So you can see they're turning their head when they feel like they're at the location of the sound, clicking in space. So cursor is the only thing on the. We carried out blocks of 50 trials, and we carried that out both standing and walking on the treadmill. What we found is that there was really no difference in localization accuracy. So most folks were a little over 1 speaker off, and that included our BLAST and TBI groups. So while there was no significant difference in their accuracy, there was also no significant difference in their reaction time. Both groups were surprisingly quicker while they were walking. It was something that caught us a little bit off guard, but we think it might have something to do with the head movement that's required for this task. We also did a simple reaction time task where we asked them to identify whether they saw 1 dot or 3 dots on the screen. With that visual task, everybody looked the same as well. So audio looked the same, visual, they looked the same. So now we're progressing to an orally aided visual search. This is audio visual. So it's still that same white and pinkish noise that's playing at the speaker location, but now we're asking them to scan the visual and find the target cluster. The target cluster either has 1 or 3 dots, and they're using a game controller to let us know. The bad thing is the whole rest of the screen is filled with 2s and 4s, so it can be more challenging. It's a little bit hard to see here, but you can kind of see how the screen is filled with dots. And Leilani Ramos is good at this task. So making it more audio and visual, we saw these groups start to break apart. Controls were significantly faster than those with BLAST TBI. They had greater accuracy when they were walking in both groups. So again, that walking seems to facilitate greater performance on this task. So this will come up again later, but I'm going to switch now to our novel PTSD intervention. And this is headed up by Dr. Mike Roy at Uniformed Services University. Again, great crew of individuals. Hannah O'Malley that spoke earlier is a part of that group as well. Now this intervention was originally developed in the Netherlands. The Karen is Motec Medical, and they are out of the Netherlands. So there is a system there at Doorn where they have a military population. It was developed in the Netherlands, and it's actually called 3MDR. It's motion-assisted, multi-modular memory desensitization and reconsolidation. The studies in the Netherlands have focused on treatment-resistant PTSD, but here at the NICoE, we're looking at a population of PTSD with a history of TBI. We are collecting a series of outcome measures, and we're looking at pre-post comparisons. Another unique thing about our study is that we've randomized whether or not participants will receive the eye movement component, and that'll be described in a little bit more detail on the next slide. All participants come in for 10 sessions. There are 3 prep sessions. The first introduces the concepts of the intervention. At that time, participants are asked to bring in songs and pictures, both positive and negative, that relate to their trauma or traumas. During the second session, the therapist reviews those images and songs with the participant, and a subjective unit of discomfort is assigned for each. For the third prep session, participants are introduced to the Karen, and they go through the 3MDR application with the therapist out on the platform. They walk through the environment using a neutral image, which is a picture of a fruit basket. If they choose to continue, they then come in for 6 treatment sessions on the Karen. Upon completion of the intervention phase, they have a concluding session with Dr. Roy and the therapist to collect final outcomes and impressions of the experience. So the virtual environment itself starts by walking in a bright environment with a blue honeycomb walkway. That's a tongue twister. The environment is purposely abstract, so it's not triggering to participants. We don't want it to be like anything they've seen in the real world or anything that they might see. Once they reach a comfortable walking pace, the first song is played in its entirety. The intent of that song is to bring them back to the time of their trauma. Once the song is complete, the environment darkens and a set of doors appears in the distance. Once they reach the doors, that opens to the first of 2 hallways. At the end of the second hallway, their first picture is posted, so you can kind of see that in a great distance on the second image down. When they reach the picture, the optical flow of the environment stops, so the picture remains in front of them, even though they continue walking on the treadmill. At that time, the therapist goes through a script, asking them first to describe the picture literally, then talk about any memories associated with the image. Finally, they ask the participant how the picture makes them feel in the present. At that time, the therapist is relaying keywords and phrases to me, and I superimpose them on top of the image out on the screen. When the dialogue ends, the participant is asked to read those words aloud. The words then fade, and some participants will be asked to look at the picture for another 30 seconds, and others will be asked to carry out the eye movement task. So the bottom image, you can see this involves a red ball moving across the image. It has numbers superimposed on it, and the service member is asked to call out those numbers. We then repeat this sequence for up to 7 pictures, with the subjective unit of discomfort being recorded after each. Once all pictures have been discussed, we return to the blue honeycomb walkway, and their second song is played in its entirety in an effort to bring them back to the present. So we were going great guns pre-COVID. We had to take a little bit of a pause, but this study has resumed and is ongoing. Hopefully by the end of next week, we'll have 3 active participants. But so far, all of the participants who have completed the 6 interventions in the Karen, so anybody who came in for that prep session in the Karen and got on there has completed the intervention. Achieving 100% compliance for a PTSD study is amazing. It was not anything that I anticipated. Overall, the PCL-5 scores have decreased from 56 down to 30. Not all the participants have seen a decrease, but all have been reported feeling better about the intervention. Some could have done with a couple more sessions. So for the next phase of things, we're adding in kind of a checkpoint, see how they're doing, and if they need some additional sessions, they will continue on in the intervention for, I think, up to 3 more? Yes, check in with Hannah. Another part of kind of 3MDR that we hope to do is to pare it down. So that's where this next study comes into play. So this is kind of where everything comes together. Can the Karen system be pared down? Can we go from the physical screen to a head-mounted display? Does it need to be VR? Does it need to be AR? Can we go from the physical speaker array that we've worked so hard on with Dr. Brungart to an audio array? And how do we do this? Well, hopefully this collaboration with the Naval Health Research Center will help get us some answers. So this is a 2-phase protocol project. We've got one protocol that is active and ongoing, and that's the comparison of visual and auditory technologies for large-scale and mobile systems. I'll go on to that in a little bit. And then our second hope, this one is not yet approved. It's in the works draft. It's how do we utilize these technologies to enhance vestibular physical therapy. Again, this is a big crew of people. This is a multi-site study. Dr. Sessoms has a great team out at NHRC. They have access to the SPA Beamer Lab individuals who are very technology savvy across the street from them. We've also utilized some help from MIT and the Air Force Research Lab. There's a lot of moving parts and development that was involved in this activity. So essentially, phase one, we're trying to leverage the advances of head-mounted display technology. Our clinicians were very hesitant about VR. There were safety concerns. There were cables. You know, when an individual is in VR and they look around in the environment, they are in the environment. There are no real-world cues. We had an opportunity to work with the HoloLens. HoloLens, a little bit heavy on the face, and we're dealing with service members who have traumatic brain injury, and a lot of them also have cervical spine issues. So again, a concern for the clinicians. More recently, about a year ago, we were able to get a Magic Leap. This is an augmented reality device you can see in the lower image there, which is a little bit low profile. It's kind of like a funky pair of glasses. I often comment that Kyle, our programmer, looks like a mole person while he's got it on his programming. But it was the first system I could get our clinicians to buy into. In fact, our physical therapist, one of which who has vestibular issues, came in and recently tried some of the applications we've developed and is very positive about moving forward with the study. So phase one is really trying to look at the Karen visual out on the screen versus this augmented reality visual in the Magic Leap. We're also looking at the physical speaker array, the audio that we get through the screen versus a virtual array that is given on a pair of earbuds. The second phase of things is to hope that if these things are comparable and everybody is happy and is giving feedback, that things can move forward to vestibular physical therapy. We want to determine whether or not how much potentially the Karen helps add to things, how much AR adds to things, or are we doing the same amount of work as conventional standard of care methods? We have yet to do that. So for the VR comparison, again, it's to compare the visual and audio components of the systems. So we want to know if they're similar. Do they give sufficient detail? Do they give feedback to the user in a different way? And then we've also developed some additional virtual environments that we can essentially put a battery of applications together so we can do a comparison Karen versus the Magic Leap. And again, getting feedback from the user, checking the outcomes, seeing how different things look. This created a lot of work, and we've had a lot of great people involved in the development phases of things. The D-Flow software, which is native to the Karen framework, does not play nice with others. So all of the virtual environments we've been using had to be recreated in Unity 3D, which is a game engine. This allows the same virtual environment to be ported to the Karen, to be ported to the Magic Leap, and to any other potential devices that come down the pike. We had to build a computer, and we had a switch installed where my projectors are now connected so I can switch between the Karen D-Flow software and switch over to Unity. So you can see the top is what the shark hunt looks like in D-Flow. I am often told that my graphics look a little bit 90s-esque, and that's fine because I am of that era. The shark hunt below, you can see with Unity, physics are a little bit better. The waves look more realistic. So we're hoping that that's an improvement as well. The Magic Leap, while it's a lot lower profile and everybody's excited about it, it had its own challenges. We had to figure out a way that we could anchor it to different locations so that the virtual environment didn't move when you moved. It could be locked right in front of you. You turn your head and you could come back to it. So that mosaic you see in the center of the screen, that's Vuforia technology. Working with the group at NHRC, we really were able to leverage that Vuforia to lock in so that we could anchor to the center of the screen. So for this protocol where we're doing the comparison, everybody is going out on the Karen platform. So the Magic Leap device will be put on them. That Karen screen will essentially be a background so that we can lock to that. As you can see with the two soccer ball applications, the one is the view, the top view is through the Magic Leap, while the lower view is actually a really cool, fun, didn't know it was taken of me picture of me doing the soccer balls on the Karen screen. So again, trying to keep things similar between the two devices so that we can really look at the difference. Okay, so these videos are going to give you an idea of how the shark hunt looks out on the Karen versus through the Magic Leap. The Magic Leap, you'll see in the background kind of the mirror, the panes of my door, because he's facing the door to the Karen space, and occasionally you'll see my wall. So it's kind of digital, virtual reality based, but augmented reality implemented. So the objective, again, they're controlling the movements. You can see on the Karen individual, those are reflective markers. We're leveraging the motion capture system to control the boat there. For the Magic Leap version, we're actually able to leverage the game controller that is put in the center of the chest, and that's allowing us to track movement. So we've had to do a lot of extensive work to make sure movements with one device are the same as the other. So if you step a foot on the Karen and you step a foot in the Magic Leap, you move the boat the same amount in the virtual environment. If the comparison study goes as we hope, we will be moving on to a vestibular treatment protocol. That protocol will have 3 groups. There will be a conventional therapy group where everyone gets standard of care modalities. There will be a group that has those conventional modalities with the Karen, and then there will be a group that has conventional modalities with augmented reality. So we plan to carry those out. Dr. Kathleen Delpy is a research physical therapist who we've been really gracious enough to have in our space. She's developed a great rapport with our clinical therapists, and a lot of their feedback has gone into making this protocol what it needs to be. Also working with the therapist, Dawn Bodell, out in San Diego, who is taking information from her own experience there. It will be carried out at both sites. So the objective of our research findings was really just to demonstrate the potential utility of using these virtual environments. We use them actively. We've been doing clinical care on the Karen for almost 13 years now between the different sites with different patient populations. Here we've seen some real benefit to our TBI and especially helping our clinicians sometimes with that PTSD component of things. We've achieved funding to allow us to pare down the system, and because we're able to pare this system down with the grant from San Diego, there's hope that there will be utility in other things. So can we take this PTSD research that we're doing and also pare that down to a head-mounted display? There's also been extensive research with the spatial audio with the headphones. Chris Montaigne is going to be doing some extensive analyses of the comparison project because, again, we are the only ones that have the physical space for speakers behind our screen, but if we can get really good spatial audio, we can provide that to the other Karen sites, and then that could also be done in clinical settings as well. So the goal is, and what I found to be very helpful, is you have to develop partnerships. If research is done in a silo, you're really missing out on things. You need to seek funding to kind of test and push things forward, and also to get staff in your space to help you. But the goal is can we learn things here where we have a lot of bells and whistles? Can we create these portable systems which allow us to deploy the virtual environments in a different way? Can we get them into maybe the clinics at the Intrepid Spirit Centers, maybe down the road through additional funding, or potentially even get them to theater to be able to allow clinicians to make assessments? With the audio-visual application I showed you with the dots, that's less than 5-minute task. If we can get that on a portable system, put it in theater, 2 individuals are next to each other in a blast. If one looks more like healthy control, maybe they can go back to duty tomorrow. If somebody looks more like a blast TBI individual, maybe they need to be reevaluated in 24 hours or seek additional care at Longitudinal. That is it. I tried, guys, to get it done in the amount of time I had. This is just some information about different publications we have and my email address and office phone number. Feel free to reach out if you have any questions or if I can point you in the right direction for anything.
Good afternoon, everyone. I think I'm live again. If I could echo something that Ms. Kruger said at the tail end of her terrific presentation that I resonate strongly with. I think to move the scientific enterprise forward in TBI and other areas, it's going to take all of us in collaboration and in partnership. And I firmly believe that. And to that end, I'll reiterate something that I said earlier, and that is I wonder what next year's fair might look like, how we might expand out on this concept, and to that end, I would welcome really any feedback, positive, negative, or in between, about how today went, ways to make it even better. So I look forward to those comments. Please feel free to send them to me directly or to Ms. Jasmine Moxley, who is still on the line, and I think everyone should have her email address. I'd like to do a final round of appreciation for our plenary speaker, Dr. Chatterjee, our range of presenters for posters, abstracts, and everything in between, and for Ms. Sarah Kruger. Thank you, everyone, for participating, and I look forward to next year. Take care.